Hello, my presentation is entitled Is there a role of the urethra in spinal cord injury-induced urinary dysfunction? Evidence of histological and innervation rearrangements in the female rat urethra. The micturition is controlled by the synchronized activity of the bladder and the urethra. This coordination is mediated by a complex neuronal network involving neurons in the lower urinary tract, the peripheral ganglia, spinal cord, and the brain. During storage, the bladder is relaxed and the urethral sphincter is contracted, permitting urine accumulation inside the bladder. These are controlled by sympathetic outflow in the hypogastric nerve to the bladder outlets and abundant outflow to the external urethral sphincter. During voiding, these mechanisms will be switched off. The bladder contracts and urethral sphincter relaxes, permitting urine to flow. These are mediated by the activation of the parasympathetic outflow in the bladder. While micturition strongly depends on supraspinal centers, it was recently proposed that input generated in the urethra may be relevant for proper voiding. These are some examples. In cats, the electrical stimulation of the urethra triggered both inhibition and activation of the bladder. In ewes and humans, the urine flow along the urethra evokes stronger bladder contractions that are abolished when the flow terminates. Finally, the urethral anesthesia provoked weaker bladder contractions, causing incomplete bladder emptying. This data suggests that the sensory input generated in urethra is important for voiding efficiency. Is this dependence of the micturition on fine neuronal mechanisms involving supraspinal centers that leaves the loot very sensitive to lesions affecting the nervous system, such as it is a spinal cord injury? Acute SCI is followed by the interruption of neural tracks between the brain and the structures causal to the lesion, causing sensory and motor impairments which affect enormously the voluntary voiding. The SCI-induced urinary dysfunction can be divided in two steps. The first one, spinal shock, in which the neuronal communication is absent and the bladder is areflexic. Secondly, with a time course, the neuroplastic mechanisms operating at the spinal cord mediate the emergence of an alternative micturition reflex. However, this is not efficient because it is characterized by the existence of NDO and DST. This leads to an hyperactive bladder and urinary incontinence. These neuroplastic changes are accompanied by noteworthy changes in mood organs. The alterations in the bladder are already well described, ranging from alterations in the mucosal organization and function and structural changes in the detrusor muscle. However, changes in urethra have never been assessed, and this is likely that the urethral dysfunction may be, at least in part, reflected by changes in urethral function and urethral tissues. So our study resorted to a spinal cord injury female rat model characterized by a largely incomplete spinal section at T89 levels. The experimental groups included spinal intact animals as controls and animals left to recover 1 and 4 weeks following the spinal cord injury. At each animal endpoint, bladder function was evaluated by cystometry before animal sacrification. After, the urethra was dissected and processed for immunohistochemistry and histological analysis. Bladder function was evaluated by cystometry under uretan anesthesia. Intact animals presented a normal bladder pattern with distant peaks of bladder pressure. 1 week injured animals presented a complete absence of bladder contractions, consistent with bladder areflexia. At 4 weeks, NDO was already established, as we can see by the increase of the frequency and amplitude of bladder contractions. Starting by histology, intact tracts presented a stratified epithelium with several layers, the most superficial composed by flattened cells. At 1 week, the mucosa suffered a marker disorganization, showing signs of cellular desquamation in the epithelium and blood extravasation in the lamina propria. At 4 weeks, the epithelium recovered its organization, however, the thickness of the epithelium and lamina propria were still significantly higher. To look for the integrity of the internal sphincter, the expression of smooth muscle actin was measured. Spinal intact animals presented strong muscle fibers with abundant expression of actin, the same happening 1 week after the lesion. In turn, at 4 weeks, the US presents marked decrease in the actin expression and also in the thickness of the US, pointing to the occurrence of strong muscle atrophy when NDO establishes. The evaluation of the external urethral sphincter cell morphology and collagen content showed no difference between intact and spinal cord injury animals, suggesting that spinal cord injury does not affect the integrity of the AUS. The general innervation was assessed by beta-3 tubulin immunostaining. We found no difference in mucosa innervation between intact and spinal cord injury animals, However, we found signs of denervation in urethral sphincter. In the US, the denervation occurs only 4 weeks after the lesion, but in the US, it occurs early at spinal shock. We also wanted to understand which type of neuronal fibers were mediating this denervation. We used the CRP immunostaining to look for peptidergic sensory fibers. We can see that there is a strong denervation in the mucosa and also in the external urethral sphincter. In terms of sympathetic innervation, we use the tyrosine hydroxylase marker. We can observe also that occurred innervation of these fibers only in the internal urethral sphincter 1 week after the lesion of spinal shock. The parasympathetic innervation was evaluated by VACT, and in this case we cannot see any difference in the expression of this marker in either time point and in either region of the urethra. We also analyzed neuronal sprouting, and we cannot see any difference in neuronal sprouting in the urethra after spinal cord injury. Concluding, our work showed that spinal cord injury provokes time-dependent changes in the urethra, as evidenced by the epithelial desorganization, the US atrophy, and the denervation. Our detailed analysis showed that the sensory and sympathetic tracts, but not the parasympathetic, were remarkably affected. This is an interesting result, which could not only explain the weakness of the urethral sphincter, which is mainly controlled by sympathetic fibers, but also the US atrophy, once the sympathetic neurons can produce trophic factors that may mediate the integrity of the US. Also, the decrease of CGRP expression suggests that urethral efferents likely do not contribute to terminal sensibilization of the root, as happens in the bladder. Taken together, our data suggests that alterations in urethra may be linked to spinal cord injury-induced urinary dysfunction, possibly contributing to urinary incontinence observed in spinal cord injury rats.